PowerPoint. So we mentioned carry information to the brain from sensory receptors. What are sensory receptors? Pressure, external receptors, or pressure, heat, pain. Then there's also receptors that the autonomic nervous system is involved in. All the autonomic functions, such as uh, how full your stomach is, when it's time to empty your stomach, when it's time to empty your bladder, lung function, all the automatic things that are done uh, by this, such as the digestive system and the respiratory system and the urinary system. All that has to be constantly monitored and processed with the information to lead to descending pathways, which uh, carry information, for instance, to skeletal muscles. So every nerve cell has what's called arresting potentials, which means there's negative charge, there's a excess of negative charge in the inside the cell, inside the nerve cell. It's created and maintained by a sodium potassium pump, and you can measure the voltage that the inside is minus 70 millivolts compared to the outside. So, nerve cells in particular use this difference in charge, or the difference in the amount of negative and positive particles. That's how they use that as a basis to generate nerve impulses. So if you look at a nerve cell and you have a grounding electrode and you insert an electrode in a cell, you're going to get that the inside is more negative in voltage, which means that it has an accumulation of more negative particles compared to the outside. Most of this outside of the uh, ions or charges on the outside of the neuron are sodium ions and chloride ions. Most of the ions inside the cell are positive potassium. But overall, there are numerous proteins and other substances that cannot uh, leave the cell, and uh, that's why overall the voltage inside the nerve is minus 70 millivolts. There are uh, leak channels where the movement of potassium leaves the cell and where the sodium channel goes into the cell. So there's the sodium potassium pump that is constantly maintaining the inside having more potassium and the outside having more sodium. So this takes energy. This is an active process, and it's very important. And it's the leak channels provide a constant uh, movement of ions in and out of the cell, sodium in, potassium out, and it's that allows the sodium pump to constantly regulate the amount of potassium in the cell and the amount of sodium outside the cell. So the membrane potential changes when ions move. They go through channels. The channels can be opened at different times to form a wave of change in the amount of sodium in the cell and the amount of potassium outside, and that's how we're gonna make a nerve impulse. Okay, so the whole purpose of the nerve cell is to maintain particular balance between the charges in the cytosol and the extracellular fluid. The cell membrane is selectively permeable. It controls what ions move in, what ions move out, and depending upon information from other nerves, these membrane channels can be opened and closed to create a sequence of events. So depolarization means the membrane potential becomes more positive. Sodium moves in rapidly. Then sodium channels close. Voltage-gated channels open. Potassium exit causes rapid repolarization. And refractory period is a recovery period that the nerve cell has to take a moment to reset. So here's what we're talking about. So the first thing, you can see that there are sodium ions. The channel is closed. Both channels are closed. And what happens next is the activation, the opening of the sodium channel. And that is controlled by the characteristics of each channel and its protein function and protein structure. So the initial movement of sodium into the cell, and this sodium is going to move into the cell, and that causes a depolarization. Depolarization means to go from a negative voltage to a positive voltage. Then step 3, the sodium channel has closed right here, and the potassium channel is open. Potassium leaves the cell as positive charge leaves the cell. We have returning to the more negative voltage because as positive charge leaves, the inside of the cell becomes more negative, and then all the channels are now closed again, and that causes hyperpolarization, where hyper is below the minus 70 slightly. So that's the time that the nerve has to reset. So this is a picture of a wave of change that flows down the nerve to carry an electrical signal. So there's 2 types of propagation. How does this wave move down the nerve? Because every time I'm holding my mouse here, every time I move my hand and click on the buttons, there are messages sent to certain muscles to contract. So some conduction will be without myelin, and some will be with myelin. Oops, there we go. So here we have the cell membrane. There's no myelin, so the impulse when sodium comes in, that causes a local current, and as that current moves, it opens up more sodium channels, and so that's conduction. So that's conduction with no insulation. If we have a myelinated axon, we have this insulation, and at these insulation areas, there can be no movement through this part of the membrane. So because you have these nodes, the current jumps from node to node. So when this positive sodium comes in, you can see that it's leading to local currents so that the change in membrane potential, the changes jumps from node to node. Behind the uh, initiation of the nerve impulse, you're going to have the nerve has to reset and repolarize. So the whole way that a nerve spike or a nerve impulse works is by the movement of different ions into the cell. As the voltage changes, different channels will open. So it's a sequential event of sodium in, potassium out, and that charge of sodium in leads to the depolarization and leads to repolarization with potassium. Okay, we don't have that. So the electrical signals are called action potentials. Myelinated axons are very important for vision, balance, and movement. You have to have a very quick response. Okay, let's stop there. Stop recording for a moment.